Hello guys and welcome back to Excelsior Academy. So as far as we were talking about the protein sorting to the different organelles. Okay, so today right now we are talking about the mitochondria. So here we'll discuss in the mitochondria like how its organization is, how the structure is present. Of course, the function of mitochondria will be discussed relatively less, because that will be completed in detail in biochemistry. Okay, in detail, like all the pathways, all the cycles with each mitochondrial enzymes, their structure and their function. So here it is just the gist of the story will be given, because the main important part over here to discuss is about how the proteins are targeted to the mitochondria. Okay, so mitochondria, it plays an important role in generation of metabolic energy in the form of the ATP. So this we all know, right? So it is also responsible for most of the useful energy which is derived from the breakdown of the carbohydrates and fatty acids. So we all know that even though the immediate source of energy is glucose, okay, but the maximum amount of energy can we get from the fat. Okay, which is converted to ATP by the process of oxidative phosphorylation. So at this particular point, which this we will cover in biochemistry, okay, when we'll talk about this carbohydrates, where we'll discuss this. And let me just tell you, biochemistry will be taken from the Leninger. Okay, so let's follow Leninger only, so that there will be no confusion. Okay, so this particular thing will be covered in that biochemistry sections, where we'll discuss how carbohydrates, fats, and proteins are broken down so that it is converted into the acetyl-CoA, and this acetyl-CoA enters the Krebs cycle and you get rest of the energy. Okay. Now, most mitochondrial proteins are translated on free cytosolic ribosomes, and then it is imported onto the organelle by the specific targeting signals. Now, if you remember, in the case of the nucleus, it was NLS. In the case of the ER, it was KDEL, right? And KKXX. Now, in the case of the lysosome, it was mannose-6-phosphate. Similarly, in the case of the mitochondrial, it is known as presequences. Okay, this is from 15 to 55 amino acids. This is what you need to remember. Nucleus, ER, lysosomes, mitochondria. Okay, these are the signal sequences. So as such, we don't have any specific name for it. Like जैसे कि ये क्या है, ये 4 amino acid, that's why it is easy to remember. But here it is around 15 to 55 amino acids, and it varies from protein to protein of the mitochondria. So that's why we don't have specific name for each. So it is coming under the category of pre-sequences. Okay. So the assembly of mitochondria involves proteins which is encoded by their own genome also and translated within the organelle, as well as the proteins which is encoded by the nuclear genome. So here we know that mitochondria has got its own genome, correct? So when it has got its own genome, obviously it has got its own RNA, tRNA, and the ribosomes proteins which will be translated, right? So that's why the targeting of the proteins to the mitochondria, it is going to vary from the mitochondrial genome and from the nuclear genome. Organization and function of mitochondria. So we know that mitochondria is surrounded by double membrane system. It has got both inner and outer membranes, which is separated by a intermembrane space. Okay, the inner membrane forms the cristae, which is to increase the surface area so that you can have more amounts of the complexes complex 1, 3, 4, and 5. So more amount of these complexes, it means the ETC transport, what we refer to, electron transport chain. So these are the complexes which comprises your ETC complex 1, 3, 4, and 5. Okay, so these are the integral membrane proteins which is present on the inner mitochondrial membrane. Okay, now why not complex 2? Because complex 2 is a soluble protein. It is your succinate dehydrogenase. Now, where do you see succinate dehydrogenase present? Where it has got its role? Anyone in the mitochondria? Hint is given that it is a soluble protein. Yes?
Krebs cycle?
Correct, absolutely correct. So succinate dehydrogenase is present in Krebs cycle. So it is also responsible for that, your FADH2 generation. Okay, that fumarate succinate का जो conversion होता है, there the succinate dehydrogenase is present, and from there FADH2 which is formed, it goes into your ATC. Okay, we'll see how Krebs cycle and ATC is connected. Okay, so each of these components play important distinct functional role, with the matrix and the inner membrane representing the major working compartments of mitochondria. And the matrix contains the mitochondrial genetic system. So you are going to have the enzymes which is responsible for the oxidative metabolism. Now, so this is just the overview, okay? Like what is a function of the mitochondria, though we are going to study in detail in the biochemistry, okay? So what is happening in the mitochondria? The glucose, the fat, they are all broken down. Then what happens when they undergo glycolysis and this fatty acids undergo beta oxidation, okay? So this is how we remember glucose ke liye glycolysis and fatty acids के लिए beta oxidation. So everything results in the formation of your acetyl CoA या फिर pyruvate. ठीक है. Now this pyruvate gets into the mitochondria. This enters into the mitochondria where it undergoes complete oxidation to give you CO2 and then ATP. Okay. Now where does the CO2 goes then? So this is exhale out, right? So each cell carries out glycolysis or beta oxidation depending on the requirement of energy. What happens on the complete oxidation? It gives you CO2, right? It means what? Each cell, each tissue is respiring. Respiring मतलब क्या हुआ? वो CO2 release कर रहा है. अब ये CO2 क्या होगा? Cells and tissue से release होके they will enter the blood system, right? Now in the blood system, guys, if you remember, we have talked about your anion exchange transporter, or we have talked about band 3, which is present on your RBCs. Do you remember? Or it is also known as bicarbonate chloride exchanger. So where we have said that CO2 will be taken, it will be converted into bicarbonate with the help of the enzyme carbonic anhydrase. उसके बाद ब्लड सिस्टम में आरबीसी ने इसको ये से कन्वर्ट किया. The same RBC will go and it will reach the lungs. Now in the lungs, this bicarbonate will again be converted into CO2 with the same enzyme carbonic anhydrase, and CO2 will be exhaled out. Did you get the connections of everything what we have discussed so far? Akshaya?
Yes, ma'am.
Yeah, good, cool. So this is how we interconnect everything, whatever we study. Sorry. Okay, so this involves the initial oxidation of pyruvate to acetyl CoA, then it goes to CO2 via the Krebs cycle. Now the oxidation of fatty acid will also give you acetyl CoA, which is again similarly metabolized by the Krebs cycle in the mitochondria. So the enzymes of the Krebs cycle, which is located in the matrix of mitochondria, are the main players in the oxidative breakdown of both carbohydrates and the fatty acids. ये भी हमें पता है, ठीक है? Now, this is just the overview. So outside in the cytoplasm, the glycolysis takes place, right? And the beta oxidation is happening. Then all those pyruvate, it enters. The pyruvate is get converted into the acetyl CoA. Now fatty acids through the beta oxidation, here it is entering, getting converted to acetyl CoA. Now acetyl CoA will enter your Krebs cycle where it will result in the release of your production of the reducing equivalents. NADH हो गया, FADH2 हो गया. This NADH, FADH2, they're nothing but the reducing equivalents. It means what? They are capable of giving protons. H plus दे सकते हैं, ठीक है? So this FADH2, NADH results in the transferring of the H plus from your matrix with the help of your complex 1, 2, 3, 4. What will happen? The proton gradient will be formed in the intermembrane space, right? Now this proton gradient is formed, right? What will happen then? Your complex 5 is nothing but your ATP synthase. So it is going to make sure that this proton gradient is used, right? And it will carry out this transfer. H plus फिर से आपका intermembrane space matrix में आएगा where this energy, okay, this energy, the mechanical energy, proton gradient की वजह से क्या होगा? The gamma rotor which is present between the F0 and F1 particle of ATP synthase will result in the mechanical energy. This mechanical energy will release energy which is taken up by the ADP, okay, to convert into ATP. This is the gist of the story, what happens in your mitochondria, right? So far so good, guys. Able to follow? Yes,
Yes,
Tanvi?
ma'am.
Okay, good. So same thing, this just now I have explained. Let's go through what if anything else is written. So the oxidation of acetyl-CoA is to carbon dioxide is coupled to the reduction of NAD plus and NADH and FADH2 respectively. Most of the energy derived from this oxidative metabolism is then produced by the process of oxidative phosphorylation. Okay. The high energy electrons from NADH and FADH2 are then transferred through a series of carriers in the membrane to molecular oxygen. ये भी आपको पता है. Towards the end, when this proton gradient चल रहा है, तो वहाँ पे क्या electron flow हो रहा है, ठीक है? At the end, this electron is accepted by the molecular oxygen and gets resulted into the formation of water. Okay, so this is why सेल नीड्स ऑक्सीजन. जो ऑक्सीजन हम अंदर लेते हैं, सो ये उसका फेट है फाइनली टू एक्सेप्ट द इलेक्ट्रॉन एंड गेट कन्वर्टेड इनटू द वाटर. Okay, now this is how the CO2 is being released, what we exhale out. So the energy derived from these electron transfer reaction is converted to potential energy stored in a proton gradient across the membrane, then which is used to derive ATP synthesis. Okay, so inner mitochondrial membrane is the site where ATP generation is taking place. And of course, as I said before, inner mitochondrial membrane, because of this crystal formation, it is resulting in increase of surface area. So, आपके सारे proteins which is involved in the oxidative phosphorylations are present in the inner mitochondrial membrane. Now, in addition to this, the inner mitochondrial membrane contains unusually high percentage of proteins which are involved in oxidative phosphorylation. जैसे कि अभी हमने discuss किया, ठीक है? Otherwise, the inner membrane is impermeable to most of the ions and small molecules. Okay, so it's a property which is critical to maintain the proton gradient that derives oxidative phosphorylation. Now, as I compare inner membrane to the outer membrane is highly permeable because of the presence of porins. Now, guys, do you remember what porine structures are? Anyone remembers? As I said, most of your integral membrane proteins are alpha helices, if you remember, whereas porins are the only ones which is made up of only beta sheets. Okay? Now, which forms channels and allow free diffusion of molecules smaller than about the 1000 Daltons. So the composition of intermembrane space is similar to the cytosol with respect to its ions and small molecules. Okay? So this is important to understand like how the intermembrane space composition and the cytosol composition is same. Okay. Now here you can ask then why there is a requirement of having a outer membrane in the mitochondria. Can anyone tell me? Did you get the question? Question समझ में आया? Krishita?
Yes, ma'am.
So think about it. What should be the answer?
Because outer membrane is permeable.
And because of that only the composition of intermembrane space is similar to the cytosol. Now I'm saying when it is similar to the cytosol, why there is a requirement of the outer membrane? इससे अच्छा उसकी जरूरत ही नहीं है. See, this is, let's say, your inner mitochondrial membrane. This is your, let's say, outer. So what we are saying is that this particular region, intermembrane space, इसका composition and cytosol का composition same है, similar है. We are saying similar है. Then I'm saying, अगर ये दोनों चीज similar है, तो इसकी जरूरत ही क्या है? It is like unnecessary synthesis of making outer membrane. उतना lipid, इतना phospholipid से लग रहा है, उतना protein लग रहा है, right? इससे अच्छा इसकी जरूरत ही नहीं है, हम इसी से काम चला लेते हैं.
But it has certain proteins, for example, just as you mentioned, the beta sheets, porins.
Because of these porins only the compositions are same. Let's say that इसकी जरूरत ही नहीं है, तो मुझे porins की भी जरूरत नहीं है ना. Because of the porins, the composition of intermembrane space and here outer membrane, it is becoming similar.
Ma'am,
Huh.
is it to separate the amount of protons that accumulate?
Correct, correct. So here when we were talking about ATP synthesis, right? Now we are making so much of effort, the complex 1, 2, 3, 4, they are making so much effort in making a proton gradient from matrix to here, right? Now, if it goes into the cytoplasm, what will happen? They are lost. The gradient is not formed. Did you get the point? To make the gradient, this is one of the advantage of having the outer membrane so that the gradient is formed. So यहाँ पे क्या होगा? H plus concentration high हो जाएगा, यहाँ कम हो जाएगा. So this gradient is used by your complex 5 ATP synthase in order to synthesize ATP. Everyone get it? Krishita, now did you get the answer?
Yes, ma'am. So selective permeability can also be one of the reasons because it would allow certain passage of ions, whereas
yes, of course.
Can selective permeability?
Yes, this is so what Shruti has mentioned is one of the reasons, which is the most important reason because we need proton gradient. Otherwise, the ATP synthesis won't be possible. And how from the starting we have studied like mitochondria is what? It is the powerhouse or energy powerhouse of your cell, right? So main important thing is about the gradient, and of course there are certain membrane integral membrane proteins which are specific only to the outer membrane, and we'll come to that because this topic is meant for the protein sorting, right? So we will come to that and see there are many things. For example, when we are talking about apoptosis, in future when we talk about the apoptosis, so there are certain proteins in the mitochondria outer region, okay, in which is responsible for helping in or making a we say as APAF. I don't know whether you guys remember or not, but they help, or apoptosome it forms which helps in the apoptosis. So there the proteins which is specific to outer membrane of mitochondria are playing important role. Okay, did you get it now? And later, see, when we study how mitochondria is going to get divided, there again these proteins are playing important role. Okay, we'll come to that. But did you get the point, like why we have mitochondria?
Yes, ma'am.
Okay, yeah. Okay. Now the genetic system of the mitochondria. So here you can see there are different genes which is like very specific to the mitochondria. So here you can see that they are the proteins which is mainly involved in the oxidative phosphorylation of your RNAs and the tRNAs. Oxidative phosphorylation proteins, enzymes which is related for it, right? For the ATP synthesis, and it has also its own RNA and the tRNAs. Okay. So the human mitochondrial genome encodes around 13 proteins which is involved in the ATC. The mitochondrial genes encode these proteins are transcribed and translated within the mitochondria, okay, because they have their own ribosomes and the tRNAs. Human mitochondrial DNA encodes 16S and 12S RNAs and 22 tRNAs. See, out of, we do have got so many codons, right? 64 codons are there. But for the mitochondria case, we have only 22 tRNAs. Similarly, the plant mitochondrial DNA, they also encode a third RNA for the 5S. This is which is important as part of your part B section where they ask a question. So universally, we know that UGA codes for stop codon. But in the case of the mitochondria, it codes for the tryptophan. AGA and AGG arginine, it is here stop codon. AUA is for isoleucine, here it is methionine. This is something which you need to remember. It is important. Okay. Because question कैसा आएगा कि आपने एक DNA synthesize किया या purify किया, and every time you're inserting a stop codon, it is not giving you the truncated region. It is giving you a complete polypeptide chain. So you can counteract, or you can give in your answer as a defense answer that instead of UGA acting as a stop codon, it is giving tryptophan. It means the DNA is translated not by the nuclear genome but by the mitochondrial genome. Okay, so ऐसे questions आ सकते हैं. Okay, now this is about the again ये question जो था, this has come for your interview. Okay, ये एक बार हमने interview में पूछा था. So like the DNA of the nuclear genomes, mitochondrial DNA can be altered by the mutations which are frequently deleterious to the organelle. Okay, so as you can have mutations in the nuclear genome, same way you can have mutations in the DNA, mitochondrial DNA. So all the mitochondria of the fertilized eggs are contributed by the oocyte, yani ki from the mother only, right? Rather than by the sperm, the germline mutations in the mitochondria are transmitted to the next generation by the mother. Okay, so here is one example. It is known as Leber's hereditary optic neuropathy. ये थोड़ा सा याद करना है, which leads to the blindness and can be caused by the mutations in the mitochondrial genes that encode components of the electron transport chain. Okay, so did you get the point? So we know that there is a oocyte and sperm jab aata hai, so only the genetic material of the sperm is actually being transferred, right? So if the oocyte in the fertilized egg, if the mitochondria is defective, then the zygote which forms or later becomes the baby, that particular child is gonna have this particular neuropathy. Okay, because of the mutations in the mitochondrial DNA.
Ma'am? Does it just cause blindness? Because if there's a problem with the electron transport chain, it should be affecting the whole organ or the whole person, right?
Sorry. Can you repeat your question?
The Leber's hereditary optic neuropathy, a disease that leads to blindness. So if it's the electron transport chain that is the problem, then it should be affecting all of the cells, right? Instead of just causing blindness.
No. So what happens is that, see, when this is like particular mitochondrial mutation, right? So what happens is that this is, first of all, it does not happen immediately once the offspring is born. Okay, it happens later in their adulthood. Okay, and again, it is not like what to say, पहले से ही शुरू नहीं होता है. At the adult life, when you reach around 20 or more, okay, if you have that trigger leads to the mutation in the mitochondria, which affects only the organ eye. Are you getting my point? I'm not able to explain properly. Let me try once again. See, it is a mutation in the mitochondrial DNA. Okay, but when the child is born, the child is normal. जैसे ही वो 20 या 20 साल से ऊपर जाता है, and if he does something, okay, what happens over there, एक trigger रहता है. So for your mutation also, the wrong lifestyle, or maybe it can be because of the taking alcohol or smoke or nicotine, whatever it is. So these kinds of mutagen will result into the mutation of this. Already they have got only one normal copy working. और इसमें क्या होगा, एक ही कॉपी बची जिसमें आपने ट्रिगर कर दिया है. इट इज लाइक प्रोटोऑन्कोजीन जिसको आपने ट्रिगर कर दिया है, and which becomes your cancerous. Okay, and that's how only that particular organ is getting affected.
Okay.
थोड़ा सा आइडिया मिला? Maybe I'm not able to explain it properly, but what I want to say is the person is already having it. It's just that trigger is required. So that trigger is after 20 or above age. तो उससे पहले नहीं पता चलता है कि that baby or that offspring is gonna have this condition, unless until you do the NGS analysis.
So is it like triggered by light or something? Because if it was lifestyle or anything, it would have triggered all the cells which have mitochondria, right?
Sorry, you're asking what?
Is it like something specific to the eyes? Because otherwise, all the cells which have mitochondria should have.
So see, if
triggered.
you I'll tell you. So if you know in the developmental biology, what happens is like when this all is being getting after fertilization, when the cell starts dividing, right? So what they have found is that whenever division is happening and the affected mitochondria, so it's not like that we have only one mitochondria. If you know, in one eukaryotic cell we have got around 2000 to 2500 mitochondria are present, right? But they all have their specific location. So when the division is happening, like once it gets fertilized and then they start dividing, right? They start dividing fast. 4 cell, 8 cell, they form the blastocyst, etc., etc. So during that, what they have said is that in the dev bio section, if you go through, they say, जब आपका ये होता है, so there they starts, you know, the alignment of or the genes expression, like which says that this should form the head or ये आपका tail बनना चाहिए. Okay, so there where most of the time what they have seen is these mitochondria which has got the defect, okay, those particular are aligned somewhere here, later which gets collected or which forms the organ your eyes. ठीक है, तो इसीलिए
it's about the axis.
It's sorry,
the axis formation or something like that. Right?
Yes, axis formation, correct. See,
okay, got it. I'll read about it.
Yes, sure. Please, everyone, go through it. Okay. I am also not able to give the proper explanation, but I have given like how it is happening. Okay, because we have to link everything, your mole bio, dev bio, cell bio, everything to understand this. Okay.
Okay, ma'am.
Yeah, good. I'll also make sure to read more about it so that next time when I give this explanation, it becomes more clear. So let's discuss about this. It is also known as LHO and Lawn disease. This is just the short form of Leber's hereditary optic neuropathy. Guys, please remind me also so that we can discuss this tomorrow in detail. Okay, because I tend to forget. Okay, let's move on now. So the coming to the protein import and the mitochondrial assembly. So as I mentioned, these are the signal sequences known as the pre-sequences, which is of around 15 to 55 amino acids. Okay, so these are required, okay, to get an information that these particular proteins are meant to be targeted to the mitochondria. अब mitochondria में भी you have to understand, एक होगा outer region के लिए, correct? एक होगा आपका inner region के लिए, एक होगा आपके intermembrane space के लिए, the proteins which is present on the intermembrane space, and the other one which is meant for the matrix. Okay, so these pre-sequences are supposed to have the information for the 4 types of the signals. Did you get it? And in this, again, 2 types of signal should be there. One for the soluble protein and one for your integral membrane proteins. एक information होता है कि अरे हाँ मुझे mitochondria जाना है. दूसरा मैं भाई soluble हूँ या integral हूँ, उस हिसाब से intermembrane, outer और inner और matrix में मुझे जाना है. ठीक है? So based on this, the transport or the import of the protein is happening in the mitochondria. Okay, let's see with the help of the figure. Okay, so now this is the example where the mitochondrial proteins, okay, they have got the pre-sequences and we're going to see like how they are getting incorporated. Okay, so this one is for your matrix. Can you see? Okay, I'll go one by one. And you can see that there are so many proteins also involved. Okay, so let's start. Okay, so as I said, cytosolic 70, cytosolic HSP 70. So what is the function? So these are your chaperones, correct? So इनका क्या काम है? To make sure that the unfolded proteins, the newly synthesized polypeptide chain remains unfolded so that later, once it reaches its target organelle, there it can undergo complete correct folding. Okay, now what do we see here? The pre-sequences is there. So now with the help of the pre-sequences, it has come to the mitochondria. Now the question comes, जैसे कि ER में हमारे पास SRP था, signal recognition particle, right? To bring it to the for your memory, for your mRNA to the ER, and that's how we started, right? But here, as such, we don't have any. We do have proteins which do recognize as the pre-sequences. It's similar to that. It's just that they have not mentioned the names of the proteins because for thousands of proteins, different 1,000 input proteins are present. Okay. Now, so at the end terminal, these pre-sequences are present. Now it is coming, it is being recruited over here. As you can see, again, ATP hydrolysis is happening. ATP hydrolysis is for what? To open your TOM complex. Okay. Now, what is TOM? Can anyone guess? It's very simple actually. The full form of TOM क्या होगा?
Something related to outer membrane.
Of course, very good. So OM is outer membrane. But इसका पूरा नाम क्या होगा? What is this?
Targeted
No.
to outer membrane.
नहीं. This is not
Transmembrane.
No, not transmembrane. ये क्या चीज है? It's a transmembrane, I know. But इसको हम क्या बोलते हैं? Like for example, in the
Transport.
नहीं, transfer भी नहीं. In the ER we talked about Sex61. वो क्या था? To which SRP was brought? I know it's a transmembrane protein, but what is the function of it? What does it do?
Transport.
Transport, it's very generic name, but when we talk about protein sorting, what is the another word instead of transport? It is your translocator, right? Sex61 क्या था आपका? Translocator था. So TOM stands for translocase of the outer membrane. So TOM आपका माइटोकॉन्ड्रिया में हो गया, सेम वे आपका टॉक होगा क्लोरोप्लास्ट में, ठीक है? So it is translocase of outer membrane of your mitochondria. अगर C आ रहा है तो इसका मतलब it is of your chloroplast. Okay, yeah. Okay, now 1 second, guys. Okay, so now so this is translocase of your outer membrane, and as you can see, to open the translocator you need always ATP hydrolysis happening, right? Now when this hydrolysis is happening, your pre-sequence goes, recognizes over here, and it enters. Now since this protein is for your matrix, so what will happen then? This TOM and the TIM, this is for inner mitochondrial membrane then, right? So they come together, okay, and then it enters. So क्या होगा? आपके इस polypeptide chain को intermembrane space जो है पास नहीं करना पड़ेगा, ये ऐसे कनेक्ट हो जाते हैं, okay? So ऐसे finally एक normal hollow बनेगा and it can enter. So from TOM to TIM23 complex it enters, and then you can see all these तो वहां पे हमने क्या देखा था? ER में signal peptidase है, right? So in the case of the mitochondria we have got the mitochondrial matrix processing peptidase enzyme. Okay, so it will remove all this pre-sequences enzyme, ATP hydrolysis फिर से चाहिए to open this, correct? Then it will come. Then what is happening again? The polypeptide chain is wrapped by the HSP70. So HSP70 ये आपका cytosol वाला था, ये आपका matrix वाला है. So it is making sure that this is not getting misfolded. Okay, now it will go to your HSP60 where the correct folding will happen. Okay, so correct folding will happen and finally it will come out and start doing the matrix related function. But if it is meant for your inner mitochondrial membrane, This will be chopped off the signal sequence, the pre-sequence, ये आपका हो जाएगा, right? And if you remember, जहाँ पे जैसे इसका signal रहेगा, the translocator, they have got this thing ना, wherever this hydrophobic region is there, they enter that lumen region of your translocator, not lumen, sorry, क्या कहते हैं, tunnel, channel, sorry, channel. So it enters the channel and then the translocator push it out. So this is how either your matrix protein will get inserted over here or the integral proteins will come into this. Okay, so this is how the protein import of the matrix or the inner mitochondrial membrane is happening. Kitna clear hua? Yes, everyone? Okay, yeah. So, Shruti, can you explain once again? Because if this is clear, then whatever is gonna come next becomes more easy. So let me see with your explanation, I will get the idea whether you guys have understood or not. Yes, Shruti.
Ma'am, like from the start, right?
Yes.
Since the so first the polypeptide comes towards the membrane, and so this Tom complex actually, if it is supposed to go straight inside into the inner membrane, then it actually combines with the TIM, so that it does not have to pass through the inner membrane, outer part of the mitochondria. It has
intermembrane space region, ये
yeah,
दोनों combine होगा तो intermembrane space.
Intermembrane space, it doesn't have to pass through the intermembrane space, and yeah, that's how it reaches the matrix of the mitochondria. If it is supposed to go, if it is supposed to enter the outer membrane of the mitochondria, then it gets pushed out sideways or by the Tom as well as TIM, depending on where it's supposed to land.
Yes, and just imagine if suppose you have a protein for intermembrane space, soluble protein here.
Yeah.
So
then it will just pass through the Tom, and then it gets, yeah.
Okay, so that is one way. That is one way that you can have your intermembrane space proteins coming. ये हो गया, ऐसा कि यहाँ हो गया, ठीक है, ये एक तरीका हो गया. Second तरीका क्या हुआ? This is suppose there, then it gets cleaved from here. तो ये आपका soluble protein हो जाता है, and this will go for your degradation. This is second method. Third method क्या हुआ कि यहाँ पे आपका signal sequence, sorry, integral membrane protein आएगा, and here you will have something. This will get chopped off here. So this is how the third time the soluble protein will come in the intermembrane space. This will go for the degradation. So there are three ways by which intermembrane space the soluble protein can be imported. Okay. Let's see if I have the figure or not. So this is for the inner membrane. So as you can see, again HSP70 is there, ATP hydrolysis hoga, TOM hoga, it will come here, right? Then for integral membrane space, you have got the TIM22 complex. Then you can see how over here it is coming, it gets formed over here, right? And we have got different translocase, OXA1, if it is a single one. अब आपको यहाँ पे ये सब याद रखना होगा to understand the difference also. I'll just write it for you. See, if it is single spanning membrane, then it goes to your OXA1 translocase. Okay. Now, what is happening? And suppose it is so many alpha helices membrane spanning are there, then it is your TIM22. Can you see it? It is TIM22. Okay. Let's go one by one. Tabhi clear hoga. So HSP70, it goes and binds to your newly synthesized polypeptide chain. It reaches your TOM complex. ATP hydrolysis is required to open the TOM complex. Now from the TOM complex, it goes to your TIM22. पहले हमने कौन सा देखा था? 23 था. अभी क्या है आपका? TIM22 है, right? Now there is addition of these proteins, TIM9 and 10. What are these? Again, they are chaperones. So they are the chaperones, mobile chaperones of your intermembrane space. What their function is, if they are not coming together, Tom and Tim साथ में नहीं आ रहे हैं, so these proteins have to pass through the intermembrane space का environment. So they do not get unfolded, misfolded or lost in the intermembrane space, right? So to make sure that TIM9 and TIM10 is coming together, bringing these polypeptide chain to the TIM22 complex, where it gets inserted or it comes in the cytoplasm. Okay? This is one way. Where, sorry, not cytoplasm, sorry, in the inner membrane space, where it will form its all start-stop sequences and it will be pushed into the inner mitochondrial membrane. Is this much part clear?
Yes.
Yeah? Okay.
Yes, ma'am.
Okay. Now, what does the second part say? So the second part, what is happening here is that, see, you have got the mitochondrial proteins also. So we know that there are so many mitochondrial proteins which is present in the inner membrane space. So the mitochondrial genome will give its own mRNA. So mitochondrial ribosome hoga, wo apna protein dega. So that protein will be again, bound with the HSP70. Okay. So that it is remained in the unfolded state. Now this will be taken to your OXA1 translocase and it will be pushed to form your alpha helices single membrane spanning integral protein. ठीक है? ये था आपका TIM22 जो ऐसे प्रोटीन आ रहा था, OXA1 हो गया जो आपका मैट्रिक्स से आ रहा था. So can you see, in both the cases it was both the proteins which is meant for your inner membrane, inner mitochondrial membrane. Now the question can be like TIM22 जो है वो आपका न्यूक्लियर प्रोटीन को हेल्प करता है, OXA1 ट्रांसलोकेज आपको माइटोकॉन्ड्रियल प्रोटीन को हेल्प करता है. Now did you get it? Yes. Okay, Krishita, would you like to try explaining?
Yes, so there is this protein which takes the assistance of HSP70. Then it would be targeted inside. I mean, when it is targeted, there is going to be hydrolysis of this ATP, and for it to get inside, it has to align. There has to be two TIM, that is TIM9 and TIM10, which are present in the intermembranous space. Then there is going to be TIM22 complex, which is going to allow that protein in. Once it gets in, depending upon whether it is a single protein or whether it is a multi. So single protein would require an essence of HSP70 because once it gets into the matrix, it shouldn't be folded. So this HSP70 makes sure that it is unfolded, and then it would be directed into OXA1 translocase, and that is how it is pushed into the inner membrane space. Whereas TIM22 complex, it would directly push those multi-traversing protein into that inner membrane space.
Okay, so that's correct, but there is a difference. See, that's why I made it like this. So this is meant for your nuclear protein, जो न्यूक्लियर जीनोम से सिंथेसाइज होकर आएगा, ठीक है. Now OXA1 translocase is meant for your mitochondrial protein. Okay, this is the difference. So ये आपका ऐसे आ रहा है, ये आपका ऐसे आ रहा है from the outside. Now this protein targeting is from the within of the mitochondria. So mitochondrial genome से mitochondrial protein आ रहा है, and then with the help of the OXA1 translocase, it is pushed into the inner mitochondrial membrane. Now it is clear?
Yes.
Yes. Okay. Yeah. So now coming to the next one. Who would like to try it as same? This is for your outer membrane porin ka targeting. Who would like to try? Tanvi, would you like to try before I explain? Would you like to try?
Okay, ma'am. Ma'am, 2 minutes, okay? Just wait. Okay, so the same, that polypeptide, to the polypeptide chain HSP70 binds, and then on the outer membrane MIM1 translocate is present, which pushes it in the outer membrane, and the other side Tom complex is present, and this whole chain passes into the intermembrane space and then folded. Okay, so it get folded, and the other side TIM9 and TIM10 is present, so that it remain unfolded. And then these then this chain goes up to SAM complex, which pushes it to outer membrane and form beta barrel proteins.
Okay, not bad, good. So little addition to this. So this much is clear, right? So the polypeptide chain is brought with the help of the HSP70 and it is kept in the unfolded state, right? Now, if this protein is meant for outer membrane, okay, this is alpha helices, okay? So it will enter the translocator MIM1, okay? If it is single transmembrane protein, which is meant for your outer membrane. This much is clear, ye itna hi hai. If it is single transmembrane protein, outer membrane ke liye hai, so it will go through your MIM1. Okay. Now, coming to the next. If it is going inside, then this protein will pass through the Tom complex. If it is meant for your intermembrane space, it will get folded with the help of the chaperones. But if it is meant for your outer membrane integration, then TIM9, TIM10 kya hai aapke? They are your mobile chaperones, which is helping them to keep unfolded state, to keep them in the unfolded state. Now, TIM9, TIM10 are keeping this protein into the unfolded state, takes it and SAM complex. What is SAM is? SAM kya hai yahan pe aapka? Sorting and assembly machinery. So if you see here, we have got 3 subunits present here. 1, 2, and 3. So there are 3 proteins. Okay, they together with the help of this, they help in actually folding so that only beta barrel is formed or beta sheet is formed. Okay. Eventually, what is happening? The beta barrel proteins, the complex is formed and it is pushed out. Now, can you tell me this mechanism is for what? It is for your porins, correct? Just now we discussed that in the outer membrane, we have got the porins. And this is how the porins are inserted into the outer membrane. What is SAM here? It is your sorting and assembly machinery. Let me just write it over here. The sorting and the assembly machinery. Now, जब भी हम कोई ऐसा meaning देते हैं, of course it has got its another name just for the layman language to understand. इसको फिर हम sorting and assembly machinery कहते हैं. Of course, it has got some other names by which it has been, you know, present or it sequence or it is present in the database. वहाँ पे कुछ और नाम रहेगा. But for our better understanding, we call it as SAM machinery, or you know, the sorting and the assembly machinery. Yes. Was it clear?
Why does this membrane have MIM? That is inner membrane, right? So mitochondrial inner membrane.
No, this is outer membrane. This we are targeting outer membrane.
There's this MIM1 near HSP70. So why is it named as MIM1?
Okay, why it is named? Okay, so for this classification nomenclature, Krishita, there is like कोई आंसर नहीं. जिस साइंटिस्ट ने पहली बार जो ढूंढा उसने कभी अपने नाम पे रखा. So they all, you know, they keep this name based on their what to say, you know, with their understanding and with their easy reference. कल को अगर मैंने कोई प्रोटीन डिस्कवर किया होता तो मैंने मेरे नाम पे रखा होता. And you won't believe I did that. I mean, of course, I didn't keep the name of the protein on my name. So when I was working during my PhD, so I was working on lectins, okay. So एक मेरे protein का नाम दिल था. So I named it like that. Why? Now everyone was laughing whenever I used to present this thing. Okay. So how I named it, since it was purified from Dhatura inoxia, it was a lectin. तो वो lectin कर दिया मैंने. Dhatura inoxia lectin. उसका molecular weight 9 था. तो मैंने मेरे प्रोटीन का नाम क्या रख दिया फिर? Dil9. So it was first time reported by me. So I kept it like that. So now it is known as Dil9. Okay. So that's how people name it, according to their how easy it is for them. ऐसे कुछ ये strong reason नहीं है कि ये MIM होना चाहिए या MOM होना चाहिए. It's nothing like that.
So the full form isn't intermembrane, not like in a.
It will be something mitochondrial import
membrane
protein
is not
membrane.
MIM.
Sorry.
Okay.
You were saying something. वो कुछ होगा, mitochondrial inner membrane हो सकता है. I'm just guessing. Okay, nowhere it is written. Okay, I'm just guessing. It might be mitochondrial inner membrane 1. But when you go in the databases, okay, the protein databases में आप जब जाते हो तो ये nomenclature use नहीं होता है. They have got a specific names for it. Okay, so for which we should know the names of the scientists who have discovered. अब HSP 70 क्यों है? क्योंकि 70 किलो डाल्टन है वो. TIM 10 क्यों होंगे? I'm sure this will be because of the molecular weight of your this protein कि वो 9 किलो डाल्टन 10 किलो डाल्टन होगा. This will be one reason. और second reason ये हो सकता है जब it was discovered, okay, intermembrane space में जब protein आपके discover हो रहे थे, they might be the 9th and the 10th protein. ऐसा भी होता है. Yeah. So yes, I'll be stopping here now. Any further doubts in today's lecture? I'll just stop the recording.